Okay, good afternoon, everybody. We're going to conclude our discussion about viral disease this afternoon. Next week, we'll move on to bacterial. And I just want to tell you all, some of you will have heard at least part of this class that I'm recording, others won't. Those of you who have heard it, well, you can use it for revision, or you can fast forward to the bits that you haven't covered yet. So we're talking initially about respiratory illness caused by virus. And first we discussed the upper respiratory tract, things like the common cold. And we moved on to discussions about viral infections of the lower respiratory tract, which are potentially far more serious because they can involve lung tissue. And this can result in very serious morbidity. So we were talking about probably the most important of all human diseases in terms of the numbers of people who are infected yearly, but also in the very, very high mortality that is sometimes associated with it, and that is influenza. Influenza is a very mysterious group of diseases which very often have a zoonotic origin. That is, they originate in animal populations, pass to the human population, where they become adapted and can be spread person to person. So I finished off with this slide, so I'll just revise it for you. And to remind you that there are two types of evolution of the influenza virus that we observe. The first is a slow year-to-year accumulation of small mutations. And these mainly affect genes which govern the structure of the H and N spikes on the outside of the virus. And the slow accumulation of these mutations as they pass through different populations in the world result in strains of the virus which can no longer be easily recognized by the immune system, even if the immune system had seen virus, the influenza virus like the year before. The immune system no longer easily recognizes the new strain. And this is the reason why we have flu vaccinations every year. What is done by the scientific community is that flu epidemics in different parts of the world are watched very, very carefully to see what their morbidity and mortality is. And those strains are characterized, and they make a decision about which three, sometimes it's two, but often it's three strains, are most likely to begin forming pandemic spread. And those strains that are selected are then passed through the virus, through the vaccine programs to produce the new flu vaccines for the next year. It is true that from year to year, we may have a small amount of protection from previous year's exposure or vaccination, but the changes are sufficient that quite serious illness can still result from the new strains. And this slow accumulation of mutation is called antigenic drift. And that is distinguished from antigenic shift. And antigenic shift is a potentially far more serious situation because antigenic shift has the potential to produce entirely new species, if you like. Although we can't really talk about species in viruses, but it gives you an idea of the degree of difference. Entirely new biologically behaving virus. And the antigenic shift is the result of the characteristic of influenza viruses that they can inhabit three different reservoirs. And these are avian reservoirs, especially birds which are associated with humans, such as pigs, such as ducks and chickens. They are both of those, ducks and chickens, are a very common host of influenza. Pigs are the other reservoir, and then humans. And each of these has their own influenza virus. Most important in the development of antigenic shift are viruses usually associated with birds and humans and their interaction either directly or with pigs. So avian influenza virus can directly jump into the human population. And if infection results and it is sufficiently adapted in humans that it begins to be spread from human to human, these form the basis of some of the greatest influenza outbreaks, the most dangerous with the highest mortality. There is another way that this can happen, however, and that is that avian virus can pass through pigs, be adapted in pigs, and then pass to humans. But there is a third way, and this is potentially one of the most dangerous, and that is that pigs can act as a reassortment vessel. So avian influenza can infect pigs at the same time as human can infect pigs. And I should also mention that pigs have their own influenza, and it has rarely, but it has occurred, it rarely occurs, but it has done, that pig virus is involved at the same time as either one of these or both. Remembering that the influenza genome consists of 8 little portions, like mini viruses. Those can get reassorted in the pig when the new viral particles are produced. And so we can get a combination of avian and human. We could get a combination of avian and pig. We could get a combination, potentially get a combination of human and pig, human and avian in the pig, producing entirely new species of virus. Though that is such a major antigenic change, it has pandemic potential because no one will have any resistance to it. It does depend on the genetics of the particular influenza virus as to how virulent it is going to be. Some of these pandemics are of relatively mild influenza strains. Others have very, very serious consequences. So these are just a few of the pandemics which have occurred since about 1900. And this is called the Spanish influenza or the Great Influenza pandemic. And this is the most serious pandemic that we know of. It resulted in hundreds of millions of people dying worldwide at a time, of course, when the human population was much smaller than it is now. So this so-called Spanish influenza, I'll explain in a minute. It actually went through several waves. The first wave of the pandemic was serious, but not nearly as serious as the second wave. And the second wave took place in 1918, and it was a catastrophic pandemic. The influenza was believed to have been a direct bird to human, although some workers think that it passed through pig first. So most people believe it was direct jump, bird to human. Some others believe that it did actually pass through pigs first and was adapted to human. It was highly, highly infectious, and it produced it had a very peculiar demographic. Now, normally in most diseases, the most vulnerable populations are the very old and the very, very young, infants and old people. The Spanish flu had a quite different demographic, and that in that it struck at mostly young people. So it was people between the age of 20 to about 35 and 40. And that was where the highest mortality took place. And we believe that this is because of two things. First of all, the influenza strain was extremely virulent, and it had a high inherent mortality just from the influenza itself. Spectacular symptoms were recorded of people bleeding from the ears and the nose copiously, people drowning in their own phlegm, all of these kind of, there may have been direct effects from the virus itself, but it appears that even more deadly was an overreaction of the immune system. The influenza virus produced a violent immune response, which resulted in symptoms very similar to septic shock, catastrophic loss of blood pressure, amongst other symptoms. Now, another characteristic of the illness was that it had a very, very sudden onset. That is characteristic for influenza in general. There's very, very short no prodromal phase. People just get up feeling fine, and by lunchtime they were either mortally ill or dead. And that is believed to have, that very great rapidity of mortality is believed to have been due to the immune response more than to the virus. It was an H1N1 virus. Go back and revise what that actually means. These are references to the antigenic character of the spikes. And this was an H1N1 and is believed to have been largely due to populations of birds, contact with birds. It began, the epidemic actually began in America. And the first wave of the epidemic in America was not that serious. But this was towards the end of the First World War. And many men went overseas already carrying the influenza virus and spreading it on the ships, then spreading it through the trenches during the war. And it is believed that another adaptation or mutation took place as it was passing through the human population to produce an extremely virulent strain of the original, which was then returned to America and, of course, to all of the countries which had been fighting in the First World War at the end of the First World War in 1918. And for 1918 and then successively for several years afterwards, there were waves of this influenza which gradually diminished in virulence. It started to just form an ordinary seasonal flu. But its genetics can be traced all the way up to the 1950s and the 1960s as participating in successive pandemics, but no longer as virulent as it was when it first entered. So have a look here, H1N1 virus derived from birds, and have a look at the next pandemic here, and what you will see is that here is the H1N1, now a human virus, now only passed human to human, mostly passed human to human, I would say, sorry, which becomes reassorted with another alien virus in pigs. And this produces the so-called Asian influenza, which was a very serious pandemic, but not nearly as serious as the Spanish flu. Oh, by the way, I said I was going to say something about the name Spanish flu. It's a total misnomer. The pandemic actually the virus originated in America. but it was called the Spanish influenza because the Spanish press who publicized it. All the other press was suppressed because the war was going on. They didn't want people to know that the soldiers were ill. But the Spanish were neutral, and they continued publicizing the passage of the pandemic. At any rate, here we have in the so-called Asian influenza, realizing names are often pejorative. We have a reassortment again. The original human from this epidemic, there it is there, with another avian virus, reassorted in pigs and producing the new virus or the new pandemic. So you can see similar sort of patterns following on. You can see very often the new strains originating from birds with human virus reassorted in pigs. This is the Hong Kong flu, which was quite serious, but not nearly as serious as people had predicted it would be. And since the Hong Kong flu, which was in the 1960s, we have had very few major influenza pandemics, but we are certainly aware of the fact that one is probably waiting in the wings, and that the COVID pandemic, which has nothing to do with influenza, don't think I'm saying that, but the COVID pandemic was a good rehearsal for the onset of a truly virulent influenza pandemic with high mortality and high morbidity. This one here is interesting because this is the novel H1N1 influenza virus, and this has made its appearance here in 1977, and it was a recombination of all three: avian, swine, and human, again reassorted in pigs, but it did not produce a very virulent. It produced a pandemic, but not a very virulent one, a very low mortality associated with it. But it is an example that it can indeed happen. And this may turn out to be much more serious the next time. Okay, that leads us to our current pandemic. Totally unrelated virus. It is not related to influenza virus at all, and this is the coronavirus epidemic. Coronaviruses get their name from the spikes which appear on the outside, because in an electron micrograph it looks like a crown. So that's where the corona part comes from. Our current pandemic is caused by one called SARS-CoV-2. And it is SARS-CoV-2 because it succeeds SARS-CoV-1, which was an earlier epidemic, which was fortunately did not develop into a true pandemic, although it did occur on different continents. So the coronaviruses are extremely common viruses in animal populations, and we actually have at least 3 coronaviruses which regularly infect and cause epidemics, but which are very mild and only cause cold symptoms. Those are probably remnants of earlier coronavirus epidemics, or which passed through the human population and adapted and became mild illness because our immune systems learned how to deal with it. But it is evident that coronaviruses are present, particularly in bat populations and probably in other rodent populations as well. There are a number of clearly identified earlier coronavirus diseases, apart from the cold viruses that I told you about. But there are several which have made their appearance extremely virulent epidemics rather. Here are a couple of them. SARS-CoV-1 was an epidemic which I'll discuss in a minute, with a very high mortality rate, very, very high mortality rate. There is another one called MERS, which is Middle East Respiratory Syndrome. It has produced very limited epidemics, but again, extremely virulent. And it is acquired from camels, but it is believed that the camels acquired the coronavirus from bats. SARS-CoV-1, it is unclear what animal acted as the vector to introduce it to human populations. It is thought to have been a food animal in Chinese markets. But again, when you examine the virus closely, it bears strong resemblance to coronaviruses, which are present in bat populations. SARS-CoV-2, again, that is our current epidemic, and SARS-CoV-2 was probably acquired also from animal populations, from animals being used as food and kept alive in food markets. Again, it bears strong resemblance to that coronavirus. So these three, SARS-CoV-1, MERS, and SARS-CoV-2, have all produced very, very virulent illness in human beings. So the first one of note is SARS-CoV-1, and which is extremely closely related to SARS-CoV-2. And it was a disease with very, very high mortality. It was 14 to 15% in younger people and 55% in people over 60. It spread person to person through, it was shown that it could be spread either through droplets, but it could also have been spread by fomite. And in the it emerged as an emerging disease in 2002 to 2003. Only 8,000 people were infected in 114 days, and 774 died. Many, many, many of them, unfortunately, were healthcare workers working with patients before the disease was clearly characterized. So they had no knowledge that it was anything other than this kind of respiratory illness with pneumonia associated that they had worked with before. There was no indication for them initially that it had such great virulence. But the thing about SARS-CoV-2 was the mortality and the very, very great degree to which people became ill was so marked that it was very soon characterized, and it was realized this is a very, very virulent disease. The mortality is high and it is very easily spread. So anybody that showed symptoms was immediately isolated. And that is one of the reasons why relatively few people became infected. Its very virulence limited the amount that it spread. Nonetheless, there was a huge risk of pandemic. It originated in China, as many of these respiratory diseases do, for reasons which are quite unclear. But both coronaviruses and influenza viruses are associated with China as an origin. It's believed that its initial spread was to Hong Kong, and that then, because Hong Kong is a major financial area, people flying in and out and in and out, it very soon made its appearance elsewhere, Singapore, Beijing, and then in the United States and Canada, as well as a couple of cases which made their appearance in the United Kingdom and Europe. But as I say, it was so spectacularly virulent that people were spotted very soon. And in fact, the major mortality was not amongst people coming into hospitals with the disease as it was amongst the healthcare workers, courageous, extremely courageous healthcare workers who continue to work with them. So it was soon the pandemic was curtailed, which was a blessing because it had the potential to be a really catastrophic pandemic. However, the second wave of coronavirus came in this form, and this is SARS-CoV-2. We have no idea about the relationship of SARS-CoV-1 to SARS-CoV-2, except that they probably both originated in bats. Here's the SARS-CoV-2 viral particle, the virion, and it is an enveloped virus, and it has a capsid on the inside, which is spiral. It is a helical capsid, and it is a positive strand RNA virus. And on the outside here, you can see the membrane, the proteins, the glycoproteins rather, which comprise the spikes. And these are important because these are the elements of the virion, which our immune system sees. And it is these spikes which have provided us with the antigens to create the vaccines against this virus. Okay, so here's the capsid. On the inside of the capsid would be the RNA strand, and here on the outside is an envelope, and then the viral spikes embedded into that envelope. And those viral spikes are there to bind to receptors on the cell membrane of the host cells, and it has particular affinity for one protein which is present on the cell membrane called ACE2. There is another one, there's another protein that it needs to bind to, which I'll show you in a minute. You don't need to remember the name. And the spike proteins bind to the ACE2. And what that does is it induces the host cell to form an endosome. That is, the cell membrane bows in like this, and it's going to form a bubble which is going to totally enclose the viral particle as if it was being eaten. You'll remember from biology that is how, for example, amoeba actually feeds. It forms endosomes to take in material from the outside. And that is true for all animal cells. So once the SARS virus binds to its requisite receptors, it's taken in. Here's the endosome there, around the viral particle. Now remember that this is a membrane. This envelope is like a membrane which has been derived from a previous host. So this envelope is actually very similar to the membrane that creates the endosome, and they are able to unite and form, and the viral particle, the virion, opens up to spit the capsid into the cytoplasm of the cell. And then there's a kind of division. Remember that viruses have to, first of all, replicate all the proteins they need. And then secondly, they have to replicate lots of copies of their genome for each new viral particle. Each new virion must acquire its own genome. So first of all, there is a set of processes where the genome is replicated. And remember, this is a positive strand virus, so it has to produce lots of copies of negative strand. It has to replicate itself and then use the negative strands to produce lots of copies, positive strand, for to act as genomes. The positive strands are actually also able to act as messenger RNA. That is the meaning of a positive strand. And the production of the viral proteins takes place through the endoplasmic reticulum of the host. The viral proteins are released into the cytoplasm, and then some of them are embedded into membrane, which is being produced by the endoplasmic reticulum and the Golgi apparatus. And the membrane, the new membrane, the viral envelope, unites with a vesicle like this, and the new genomes are packaged together with all the viral proteins that make up the capsid, and the endosome then makes its way, the vesicle makes its way to the cell membrane, where it unites and liberates the new viral particles. So there are various stages here which are open for chemical intervention to try and stop the reproduction of the viral particles, and these are the basis of valid medical treatments for SARS-CoV-2 infection. One that is not valid is this process here, the formation of this initial endosome, bringing the particle in. There is a similar process in the production of malaria, and ivermectin is used in treatment of malaria, not with enormous success, it has to be said. And people thought initially that this might be a way to stop SARS-CoV-2 infection, but it doesn't. Ivermectin has no effect on SARS-CoV-2. Okay, so just to mention, the vaccines which we use against SARS-CoV-2 are extremely successful in one aspect, and that is in drastically reducing the incidence of serious illness. Most people who have received vaccine may in fact acquire SARS-CoV-2, but it is usually extremely mild if they have been vaccinated. The mortality is much, much higher in people who have not been vaccinated. And quite apart from the mortality, the virulence of the disease can cause real hardship. It is not a mild illness if you haven't been vaccinated. And it can, in fact, produce long-term sequelae, like long COVID, as well as other side effects. For example, one of the cells which the virus binds to is endothelium. And endothelial cells line our blood vessels. And so for that reason, especially in people who have not been vaccinated, there is a strong association with blood clotting disorders and things like that. The vaccine that we use is an extremely cunning idea, and it has been in development long, long, long before SARS-CoV-2 was ever heard of. And the most important part of the vaccine is actually the outside. It is like a little package, and it functions a little bit like a virus in a way, because the package is designed as rather like a viral envelope. It has associated proteins which bind it to cells of people who have been vaccinated, and it will inject, if you like, the contents of that package into the cells. And what is packaged into the vaccine container, if you call it that, is messenger RNA. It is synthesized and it is translated in the host cytoplasm by the ribosomes to produce viral spike proteins. And those viral spike proteins are soluble. They are released from the cell, and the immune system responds to them and recognizes them as an antigen and effectively learns what the spike proteins of the virus look like. And that is how the vaccine works. As I say, the vaccine is extremely successful, but not so much in entirely stopping or preventing acquisition of the illness, but in drastically reducing its virulence. Okay, so I just mentioned one other virus which is of importance, especially now as we approach winter. This is another one like influenza and cold viruses. They become more prevalent during winter because it's of the cold and the dry air. They actually are able to last for longer as droplets in the air. So the droplets last longer and the virus is more easily transmitted, especially because we tend to crowd into places. We don't spend nearly as much time outside during winter. So this is respiratory syncytial virus. And just as a note, respiratory syncytial virus, it's an envelope virus, but it is a negative strand RNA virus. In adults, it's usually limited to the upper respiratory tract, although it can produce severe bronchitis. But in children, it can be extremely dangerous. And in young children and infants, it can form a lower respiratory tract infection, which results in violent bronchitis, bronchiolitis, which is inflammation of this very small air passageways in the lung, and also a pneumonia. And quite apart from the fact that it predisposes to bacterial infection, it can in children produce its own viral pneumonia, an inflammation of the lung tissue, which results in fluids becoming accumulated in the alveoli, which get consolidated and obviously impair breathing. For this reason, it is a serious illness in children. There are very effective vaccines that are available. And not only children should have it, but adults should have it as well. I had this at the beginning of the year, and it is an extremely unpleasant thing to go through. Okay, let's talk about some of the other important viruses in humans. And this is a group of viruses which you have surely heard about. And these are the herpes viruses. The herpes viruses are extremely diverse. They all have one characteristic, which almost all of them show, and that is that they can enter latency. And that latency is very often within nerve cells, even though their manifestation may be elsewhere, very often on the skin or mucous membranes. They tend, many of them, to go into latency by integrating with nerve cell genome. So the first group of the herpes simplex viruses, or abbreviated HSV, and 1 and 2 are the most important. So herpes simplex 1 is the common cold sore or oral herpes virus. That is usually, in most people, is acquired in childhood. And it produces the characteristic lesions that I'm sure you're familiar. If you hadn't had them yourself, you've probably seen them on other people, that they produce. It produces oral lesions. Here it is here. This is a DNA virus. And this is its manifestation here. We're in the cold sore form, and it produces this lesion of blisters, which are sensitive, painful, and last usually about 10 days before they resolve completely. And these blisters are produced sporadically in childhood, maybe every couple of months or whatever. And then in adulthood, honestly, there can be intervening years between them appearing and not appearing. And people associate them they're called cold sores because there is some indication that exposure to really cold weather may produce that, probably because it stresses the skin cells, it stresses the mucosa. But the other manifestation of HSV is HSV2. Now, they are very closely related, as I'll explain. Now, this HSV2 is the commonest cause of genital herpes. And the symptoms of this, it usually begins, there is a prodromal phase where the skin has a peculiar sensitivity, almost like sometimes an itchiness, but a sensitivity to pain. And that then develops into this fulminant infection where there are blisters, and these blisters are leaking serum, which is extremely rich in viral particles. So it's at this stage that the person becomes infectious. If they do not have open blisters like this, their infectivity is lowered, and if they have no symptoms at all, they are essentially non-infectious. In men, the appearance of the blisters is often preceded after the prodromal phase by a very pronounced urethritis. So men tend to go to the doctor with this much sooner than women because the urethritis is extremely painful. In women, the disease progresses pretty much the same through prodromal phase, etc. But often the lesions are in the vagina and they may not be noticed. It may just be felt like a yeast infection or something like that. Itchy, a nasty feeling, but not necessarily obvious that it is a genital herpes infection. There is something to mention, however. That is that genital herpes can be spread very easily through oral sex. And then there can be a confusion because you can actually acquire HSV2 as an oral infection, and you can also transmit HSV1 as a genital infection. There's little difference in the pathology of the disease. And in both cases, the disease may become latent and disappear for a while and then return. However, there has been a tremendous amount of disinformation about genital herpes. If you have a genital herpes infection, it is not the end of the world, because for two reasons. First of all, you don't know that it is definitely going to come back. You don't know if there is no sign of the infection, you are not yourself infectious. Women need to be extremely careful to determine that they are not infectious. The second thing is, at the onset of symptoms, there are good drugs now which will shorten the length of time of the episode. Valcyclovir is the commonest one. Women need to be particularly careful, and that the reason for that is if they are pregnant and they give birth while they have active lesions in the vagina, a number of things can happen. And the first is simply that the eyes become infected and they acquire keratitis, an inflammation of the cornea, which can lead to opacity of the cornea. And in addition, such instance of neonatal herpes can spread to the brain and result in neurological damage. So it is normal to care during pregnancy for an obstetrician gynecologist to examine very carefully both past history of possible herpes infections, but also to look for them to ensure that if somebody does have an infection, it's again not the end of the world, because they can be put onto valcyclovir or something similar, and also can be offered the opportunity of a cesarean to be absolutely sure they don't infect the infant. Okay, so here is another one, and that is this one here, varicella virus. And varicella zoster is its name. And this is the causative agent of chickenpox, which used to be an extremely common childhood illness. Chickenpox is extremely infectious, and is spread by direct contact, by respiratory droplets, things like that. And it results in a widespread development of these pocks. Each one of these little dots is referred to as a pock. It's an itchy rash, which can spread through the whole body and can even erupt on the mucosa of the mouth and throat. And they produce a fluid, and a pussy kind of fluid, which is very, very highly infectious. It's full of virus. These pocks are quite deep in the skin, and they can result in a secondary bacterial infection. There are some potential serious side effects, especially in children. But the most serious thing about varicella zoster is that, just like so many herpes viruses, it can become latent. And when it does so, it inhabits nerves of the body, and often in specific areas, sometimes of the face in a band, but most commonly of the trunk. And if that erupts, usually much later in life, it can erupt as shingles. And this is what typical shingles rash looks like. Now, these look relatively benign in this photograph, but they are agonizingly painful. And in fact, that pain can be so severe that people can go into shock, especially in the elderly. And they can, in addition to that, they can also cause inflammation of the nerves themselves and result in facial paralysis, in persistent pain, even after the rash is resolved. And shingles has the potential to be extremely serious in human beings. And there is a very good vaccine against shingles. In fact, there are a couple now. And it is really a good idea, even if you don't know that you've had chickenpox, it is a good idea to get the vaccine. It's essentially a chickenpox booster. Now, most of children in the United States have already been vaccinated against chickenpox. It's a very good chickenpox vaccine, and it's a very good idea to get it. If you present that initial infection, then you almost certainly will not get shingles later on, but it can occur. Unfortunately, the chickenpox vaccine is another of the vaccines which anti-vax movements have turned against. And so chickenpox incidence is rising across the United States. It's now becoming common, much more common than it has been since 1995, when the first really good vaccine was produced. I should mention that the vaccine is an attenuated live vaccine. That is one of the reasons why the vaccine movement does not want to see it used. But attenuated vaccines have been used safely for many different diseases since the days of Pasteur. And attenuation has been proven to be an effective and safe way of producing a vaccine. Okay, now just to mention a couple of other diseases that especially have skin manifestations. There are a whole group of viruses called the paramyxoviruses. And the most important one of all of these is measles. And measles is a very, very highly contagious disease. In fact, many people say it is our most contagious human disease. It's also known as rubeola. And it gets that name from the fact that the skin turns red. It produces this disseminated fine rash of red marks. These are not blisters like chickenpox. This is a true rash, like erythrogenic rash. And the disease is spread largely through respiratory droplets. And it has a characteristic that it also forms red patches along the gum line. You can just see, this is not a good picture, but you can just see them there, these little red dots here, along here. Those are conflict spots. Now, it has the potential to be extremely serious in children, and it can result in all sorts of sequelae. It can even have neurological associations, and it can also affect the eyes, in some cases, producing impaired sight. But the most dangerous thing about measles is not the immediate acute illness. It is that it forms a long-term, persistent infection. Now, it's not a latent infection. It infects the cells of the brain and forms a long-term, low-grade, chronic infection, which often people have no knowledge of until a decade or so later. So if you work it out, most children will get it when they're about 20. So what am I talking about? Most children will get it when they're about 5 to 10. And so about a decade or a decade and a half later, they can develop symptoms of this disease, subacute sclerosing panencephalitis. And the sclerosing part refers to the destruction of brain tissue. Subacute because it's not noticed for so long, and panencephalitis because it is globally affects the brain tissue, causing inflammation of brain tissue, which is encephalitis. It can also spread to the bone, in which case it is called Paget's disease. And unfortunately, subacute sclerosing panencephalitis is invariably fatal. This is a rare outcome. This is a very rare outcome, but it's such a devastating outcome that it is worth having the measles vaccine, which ensures that it will not occur. Unfortunately, this again has come under the ire of the anti-vax movement, and it's largely because of this. This is the vaccine for measles, which is a very good vaccine, is included in this vaccine combination called the measles, mumps, and rubella inoculation. And unfortunately, somebody wrote a medical paper, a totally spurious medical paper containing fake data, attempting to show that the development of autism was linked to children getting this vaccine. There is no association whatsoever. Millions upon millions have been spent showing that there is no such association. And as a result, there has been a massive movement against this vaccine, and measles is again on the resurgence throughout the world. Wherever people hear this story, this totally fallacious story, they tend to opt not to have their children vaccinated, which is a very bad idea. And this is measles. Here's the next one, and a somewhat related virus. And this is mumps. This is also called infectious parotitis. Parotitis refers to infection of the salivary glands. I'm trying to remember which ones they were, but anyway, of the salivary glands, and it produces this characteristic swelling around, sometimes called the bull's neck swelling. in children. It is a relatively mild disease. There is possibility of some, in children, of some sequelae, but this is very rare, and it usually passes off without any need for treatment or anything except bed rest. However, in adults, it can be extremely serious because the infection strikes at various different organ systems. The most spectacular, I suppose, is that it can cause a massive orchitis or swelling of the testes in human beings. And I'm talking like that size. And in males, that totally disrupts the structure of the testis and can result in infertility afterwards, quite apart from the fact that it must be exquisitely painful. But in women, by the way, it can also cause swelling of the ovaries, but the testes are bound by a fibrous coat. So the testis swells against that. It causes tremendous pressure inside the testis, which accounts for the pain, but also for a lot of the disruption of the structure of the testis. In the ovaries, they do not have that fibrous coat around them, so they can swell and go down without major damage to the ovary. But mumps virus can also cause infection of the pancreas, causing pancreatitis with the possibility of resultant diabetes. But it can also cause an encephalitis, an infection of the brain and an inflammation of the brain. So in adults, it's a very serious, can be a very serious illness. The measles, mumps, rubella vaccine is a very, very long-lasting vaccine. The immunity lasts a long time, so this is protective even in adulthood. The last one in this group, or the MMR group, is rubella. And rubella is also called German measles, for reasons I don't know. But it is, in children, an acute, mildly infectious disease. It's of no real seriousness at all. And even in adults, it's a self-limiting disease, and often is just passed off as a flu that people might notice that they have a little bit of a rash. However, that is not its true seriousness. It is really serious because in pregnant women, if they acquire rubella, then the fetus can become affected through the placenta, and the children may be born with all sorts of developmental problems, especially eyes, ears, damage to the heart, and also damage to the limbs and development of the limbs. So in pregnant women, it is very, very serious, a very serious situation if they acquire rubella. And it's for that reason the children are vaccinated with the MMR vaccine, is to so that they do not act as vectors of the disease, of the virus, to women who are pregnant. Okay, so our next along in our dreadful gallery is human papillomavirus. And I have said something about this before, and that is the human papillomavirus is a very, very common virus, has a number of different strains, but they are all somewhat interchangeable in the effects that they produce. And the one that we know best are common warts. Here they are here, very common in children. As you get older, your immune system tends to recognize the virus very easily. Another strain produces the familiar plantar warts or verrucas that people are convinced that they get from gym floors. You don't, you've already got the virus. And it just manifests itself for one reason or another. And these are not particularly, they're a nuisance, but they're not particularly serious. But and the truly serious ones are the genital warts. Now, the genital warts are large, often are large, and very, very unsightly around the genitalia, either male or female. And they cause great distress. They're extremely common, especially in young people. They are easily transmitted one to another, and they can actually be easily treated as well. That's usually self-limiting. If you leave them alone, they will go away on their own as your immune system recognizes them. But the problem with this is that the genital warts can also form a persistent infection, not latent, but a persistent infection. And it is essentially symptomless for many, many, many years. Its major manifestation is in cervical cancer. So this is a cervix here, and all of this tissue looks inflamed, and it frankly looks diseased. Is tissue which has had a chronic infection of genital warts or papillomavirus, and this acts on the regulation of cell division of the cervical cells. So this is an example of an oncogenic interaction. The papillomavirus in this case is oncogenic, and it transforms the cervical cells into cancer cells. The same thing can happen on the penis, but it is very, very much more rare. Cervical cancer is actually quite common. It's one of the commonest cancers. So that's the first thing to say. The second thing is in genital warts is that they can be transmitted to newborns and during delivery. So this is a very good idea to get the vaccine, which is called Gardasil. There are a couple of others as well now. But this vaccine is best given before the child becomes sexually active, because the virus is so common that immediately people become sexually active, they are almost certainly going to be exposed to it. So it is the best idea is to get this vaccine early, and that get your children vaccinated before they become sexually active. So just to show you the impact of this, this is an Australian study of the incidence of, the prevalence of cervical cancer. And you can see up to here, this here dotted line, it's pretty much constant. But look, this is where the vaccination against genital warts, against papillomavirus begins as a widespread public health initiative. And this here shows how the incidence of, the prevalence of cervical cancer plummets as soon as the vaccine is introduced. This is an extremely successful vaccine, an extremely successful vaccine program. Unfortunately, in the United States, it has not been nearly as successful, largely because people do not want to have their children vaccinated, because they, quite frankly, don't want to admit that their children will become sexually active. Okay, so to finish off, I'm just going to very quickly have a discussion about human immunodeficiency virus and the disease of AIDS. AIDS stands for acquired immunodeficiency syndrome. Now, the reason that I make a particular point of doing this is because information about HIV has become somewhat rare. In the late 80s and the early 90s, I was actively involved in campaigns to try and get AIDS recognized and to support people who had AIDS and everything. And following that, all of that activity, it became extremely common for information to be given out very freely at universities, schools, everything else, especially information about safer sex practices, that sort of thing. This has become unfashionable in a way. And I find, to my horror, that many, many young people actually don't know anything about HIV. So I make a point of just discussing, this is only a brief discussion, but it will give you some information at least. So HIV is a very interesting virus. It is one of those retroviruses. And I'll remind you that a retrovirus is a positive strand RNA virus, but it cannot use that positive strand in the host cell until the positive strand is converted to a DNA strand. So this is very peculiar because this is like the reverse of the central dogma. Normally we say DNA is transcribed to RNA, the messenger RNA. Now we're saying, well, here we've got a positive strand, which is like messenger RNA, and that is being reverse transcribed. It's being reverse transcribed to DNA, and that DNA strand then becomes integrated into the host genome. That is why they're called retroviruses. The retrovirus here, the structure here, this is an envelope here, and on the outside it has the characteristic spikes that we see so often. On the inside it has this conical shaped capsid here, and it actually has two strands of RNA. They've just shown one here. Here it is. Here's one of the strands here, and associated with that strand inside the capsid, there are molecules of this enzyme, reverse transcriptase, and it is reverse transcriptase that is going to copy the RNA into DNA. And they produce from the RNA, they will produce a double strand DNA strand, which then will integrate into the host genome. So these spikes on the outside recognize particular proteins, of course, on the host membrane. And these proteins are CD4, that is a receptor which is particularly important in the immune system on a group of cells called the T helper cells. But this is also present on other cells. It is present on macrophages, and it is present also on neural tissue. They also have to bind to another one, and this is called CCR5, and this is a receptor for cytokines, which are immune hormones. It has an entirely different function, separate function, but the virus recognizes it. So it has to bind to both of these, CCR5 and CD4. When it does so, the capsid, it unites with the cell membrane, and the capsid is spat in and is degraded. And the positive strand is released. The positive strand is reverse transcribed by the virus's own enzymes to a double strand, and then becomes part of the host genome in the nucleus. And there it goes silent. So it is actually present. It has the potential to become active, and a certain number of them actually do become active. So this is not a true latency. It is more like a persistency. And the way in which this is going to convert from just being part of, silently part of the nucleus of the genetic material of the host, is that this is activated when the cell is activated. Now we just think particularly about the T helper cell. The T helper cell is part of the immune response. So when that T helper cell is activated to do what it has to do in the immune response, the genes for HIV are activated at the same time. And what happens now is that they begin to be transcribed. And they will transcribe this into messenger RNA, into viral messenger RNA, which then is exported to the cytoplasm. And it does two things. First of all, it replicates the whole genome over and over again, as we've seen before. The second thing it does is it produces the viral proteins, but this is very important. It produces these viral proteins in a particular form. And that is that they are produced as a long transcript and a long translation. So the first stage in translation is we end up with a very long polypeptide, and it needs to be cut into pieces. Each cut piece forms a protein, a mature polypeptide. So the reason I'm emphasizing this is when we talk about drug interactions, drug treatments, you will hear about this again. All of these are packaged again, and then the viral capsid is taken into a bud like this and is budded out to form the new virion. So the reason I am emphasizing these different stages is because each of them represents a stage in the viral life cycle which has the potential for intervention. Initially in the AIDS epidemic, this was a common site of interest, stopping this process from taking place. It didn't really work. The second site of interest in research was this stage here, where the viral RNA is reverse transcribed into DNA. Now, this produced the very earliest drug intervention, which was widely accepted and widely used. And this was to use drugs which interfered with the transcription, with the reverse transcription. They stopped reverse transcription. These drugs do definitely have a certain efficacy. And one of them that you may have heard of is azithromycin, although there are a number which operate in a similar way. Azithromycin interferes with reverse transcription. The problem with azithromycin is, first of all, it had very serious side effects sometimes. And second of all, it was shown that the virus very rapidly developed resistance and developed strategies to evade the effects of azithromycin. But azithromycin is still a very important drug. In many developing countries, it's all they have. But it also has a very great efficacy as a prophylactic. If somebody has been exposed to the virus, then you can administer azithromycin immediately or as soon as you can. It can prevent an infection. So it is very often, for example, it is very often given to pregnant women who are giving birth, who have HIV, to prevent transmission to the child, because that transmission is usually during the birth process. In other people, maybe have made a mistake when they're having sex and they didn't have safe sex. You can take azithromycin for a relatively short time, like 10 days or so, and be relatively sure you won't get infected. So it does definitely have its uses, but as a long-term treatment, it is useless. But returning to this here, where they had a long strand, we had a long strand of polypeptide produced, which needed to be cut into pieces. When you cut a protein, you use an enzyme called a protease. And a group of drugs were developed which are used to inhibit the proteases from operating, and they are spectacularly successful. I'll tell you on a personal basis, I had friends who were literally on their deathbeds and had no treatment other than things like azithromycin, which we knew was useless. And it was a terrible, terrible thing to see young healthy, otherwise healthy people, reduced to skeletons lying in a bed waiting to die, who were then given protease inhibitors and recovered within weeks and then completely within months. They are so effective, in fact, that people's circulating virus, the amount of virus they produce in their bloodstream, is reduced to zero. People on these protease inhibitors return to a state where they are completely uninfectious. Unfortunately, they are not cured. This is a persistent infection. We don't exactly know how, but if people, if you remove protease inhibitors, unfortunately, the disease very soon reappears. So there are limitations. So the disease of AIDS is the disease which results from the collapse of the immune system. As the virus begins to reproduce in activated T cells, it actually kills them. It eventually kills them. They produce about 350 viral particles and then die. And they are not rapidly replaced. And as a result, the number of T cells, T helper cells in the body begins to decline. So we'll have a look at this graph. First of all, here is the number of CD4 T cells, the helper cell, T helper cells in the blood. And this is cells per microliter, which is the standard measure. So the very first thing we're going to look at is the amount of virus that is present. So here's an uninfected person just being infected. Within a very short time, they suffer an acute HIV infection, a very, very acute infection. And often people feel as if they have a terrible, terrible flu. And this is very early on, long before probably they even suspect that they have HIV. They just think they've got something else. This is in public health. This is a very important thing because look at the level of the virus. They have very, very high virus titer, very high viral count. In fact, it is high as it ever will be. Even in the disease, it's about the same. In between, it's not nearly that level. In public health, this is important because people in this area here have no idea that they have the virus, but they may be having sex or exchanging needles or doing something like that and are very, very infectious. So soon after that, the immune system initially recognizes the presence of the virus and combats it and reduces the numbers spectacularly, but they don't reduce it to zero. Instead, the virus persists as a persistent infection, and this can be for a very long time. This can be for 5, 10 years that the virus will persist, and it will slowly increase. And as it's increasing, remember, its primary host cell is the T helper cell, part of the immune system. So let's look at the T helper cell count. Well, normal T helper cell count is about 1,200 cells per microliter. Here it is here. The initial infection results in a drop in number, but the number rebounds as the viral count drops. So there's something of a recovery of the immune system. But as this infection persists over the years, the T helper cell count begins to drop. It's at this stage here that people begin to suffer from strange infections. They start to have persistent fungal infections, for example, where before if they had a fungal infection, they would recover from it very easily. Not major things. This is almost like a prodromal phase. But the symptoms begin to increase in severity as the immune system begins to lose its efficacy. So around about this stage here, that people will notice may report with very rare diseases, which normally do not occur in younger people or do not occur in their demographic. They may even appear with strange zoonotic diseases, diseases from pigeons and things like this. Their immune system is beginning to collapse completely and failing to protect them, even from things which are normally to our immune system perfectly obvious and very rapidly removed. At this stage here, where the CD4 count drops to less than 200 per microliter, this is the point at which a clinical definition of AIDS is given. And there are certain diseases which I'll mention in a minute, which are characteristic of this period here. Without treatment, without protease inhibitors or anything else, death will follow probably within months. And the death will occur not due to the virus itself, but due to these secondary infections, due to the infections which result from the loss of the immune system itself. And at that stage, the level HIV is extremely high, about on par with what it was in this initial infection. So that's the sad course of the disease of AIDS as it used to be. Nowadays, that is in developed countries, at least in the United States, Europe, etc. This is not the course of the disease. And people infected with HIV have every hope of living perfectly normal lifespan with the use of the protease inhibitors. Sorry, my screen is frozen again. Okay, I'm just going to, we can just finish off with this as my computer is sulking again. But let me just emphasize for you. The reason that I give this little talk on HIV and AIDS is HIV is very, very much with us. It is quite common within the gay population, within drug-using populations still, but worldwide, it is a disease of heterosexuals, not just of the homosexual gay community. And it is still something which anybody who is sexually active with more than one partner who has not been shown to have AIDS, have HIV, anybody who is sexually active needs to think about the fact that HIV is out there. Now, there are different strategies that one can adopt, that being the case. The first and the simplest and easiest is to adopt safer sex practices. And the safest sex practices are the most effective one is to use condoms with each and every sexual encounter, no matter what that sexual encounter entails, because you never know what sexual encounters are going to entail. That's something that needs to be admitted. Sorry, my computer is really just, oh, here you go, sorry. Let me, I'll just do this slide and then go back a bit. So that is number one. Number two, if you're an intravenous drug user, you must assiduously use your own apparatus, your own needles, etc., and never, never, never share, because this has been one of the major ways in which HIV has been transmitted. HIV is always transmitted by body fluid. You cannot get HIV by droplet inhalation. You cannot get it from touching somebody, hugging them. You can't even get it if you kiss them. And even oral sex, although it does have a risk, is a relatively low risk compared to inserted sex or exchanging needles. So it is true that the one sad way that HIV can be transmitted is during pregnancy and during breastfeeding. So a mother who is infected with HIV can in fact transmit to an infant, either in the womb or during breastfeeding. So the prevention entails recognizing all of the risks of the behavior you are engaging in. The condoms are probably the easiest obtained and used prevention method. But for those of you who are who believe that you are at particular risk for one reason or another, maybe you're gay and you're having an active sex life, or whatever, there are prophylactic drug regimes which you can now use. And the commonest one is called PrEP, pre-exposure prophylaxis, which is a very low dose of very effective anti-HIV medications. Very, very low side effects to these. There are some which they will mention to you if you ask for them. And if you believe that you are in a high risk group, you need to go and talk to somebody, talk to an authority on the issue, and decide whether PrEP is actually something that you can accommodate and use. It is extremely effective. Now, the treatments that I have actually told you, azidothymidine, which is used prophylactically if people have been exposed, protease inhibitors, which inhibit the viral proteases and are extremely effective and reduce people's viral load to zero so that they are themselves not infected. Then there is another set of treatments which is related to this. Remember, I told you that azidothymidine, one of the problems with it is that the virus soon develops resistance. The same might be true of protease inhibitors, but what they do is there are a number of protease inhibitors, and in highly active antiretroviral treatment, they use a combination of drugs which they continually change, so that the virus never has time to develop resistance to any one of them. And this is proved to be extremely effective. There's a continual search for vaccines, but the vaccines have never been particularly successful. We have no animal model. We only have human. HIV is a strictly human disease, even though early in its evolution, it was probably a zoonotic disease. But we have no animal model to use to develop vaccines. So it has been extremely difficult, and no successful vaccine has yet been produced. That's not to say it's not going to happen, but up to now, it hasn't, despite all the research. Okay, let me just check because I flipped around. No, I think we've covered just about everything. Okay, so thanks for sitting through this long lecture, and I will see you again next week.